is not how this is supposed to look. We're going to do our best. It's literally 2 minutes ago I signed in, everything was fine. And then Columbia said, I'm not authorized using my signing credentials to start the Zoom link. So we have to make sure that Chip Clark is able to see us and talk to us. So I'm going to have a bunch of windows here, hoping I'm going to be bouncing back and forth between them. So I ask for your indulgence. And let's get class started. Okay, welcome to week 3. It is after 9 o'clock. 9:03. Everybody sign in when they came in. Terrific. Today we're going to use as a jumping off point from, we're going to jump off from some of the things we learned the first 2 weeks, put them into place, and rope a few of you in to present. Remember, presenting is not a evaluatory thing, it's just as a springboard to learning how to do this. So, it's not a matter of doing well, or not doing well, just approach it fearlessly. Okay, a little housekeeping. Dropout is over. We're going to start taking attendance. Again, this is a legacy from having taught the class 11 years and making sure you all show up. Sign in and then sign out. Please don't sign for somebody else. I do check the handwriting. It just makes you look bad. The class, there have been a few people that have had legit reasons for not making it to the class occasionally. So I'm trying to record them. And I've got a little YouTube page. I go to, DV Sable is, I just sign in from that. So if you go to YouTube, you go to DV Sable, my little amateurish page will show up. And that's got a video of the class. So the first week I kind of stuck the camera on the floor, so you see a lot of backs of heads. Last week it almost looks okay. This week I'm recording it in the back there, so hopefully it'll be understandable and seeable. And use it as you need to. This is not an excuse to not show up and just watch the video later, because attendance is mandatory. And if nobody showed up, I don't want to lecture to an empty room. And that takes away some of the interactive parts of it. So, most important thing about being a founder is showing up. And we try to live that from the beginning. Today, our speaker series begins. Chip Clark is an extraordinary guy, CEO of a very interesting biotech company up in Boston. And I'm really looking forward to having him tell his story. He will be the one person in the class not wearing a mask, so he doesn't have to shriek and then be too soft the way I do. So, actually, could you close the door? Terrific. What did everyone do to prepare for class today? I'm not going to embarrass anyone by calling you out. That starts next week. What specifically stood out in your mind for the assignments and the suggestions, the reviews, or the things that hopefully you found on your own to reinforce the early entrepreneurial process, or to start giving yourself more of a grounding in the biotech industry, in biotechnology as a science, or epidemiology, or biostatistics? In the back.
I just found that all-inclusive medicine needs more diverse genomic data to be really interesting, specifically when they were talking about the
Great, it was written by one of our classmates. Next. Okay. Yeah.
When I was reading the Twitter thread, I realized I didn't know what PRS was, and that's why I was really confused. So then I went down a bit of a rabbit hole, figuring out what PRS was as statistical method.
Describe to everybody what PRS is, what its relevance the reading was.
So PRS stands for polygenic risk score. I'm going to try to do my best to explain it. But
okay, that's good. That's good. Let's leave it at that. Remember our theme. One of the overriding themes of working in biology this century is biology is where we have the rocket fuel of going analog to digital. And what fueled that rocket fuel was the genome. Okay, we elucidated the genome in 2000. Suddenly we have a Rosetta Stone to take our observation of disease and our description of disease to a much more reproducible, precise level. Okay? Now, the easy stuff we had started before the genome was elucidated. You may have heard of the Philadelphia chromosome, Peter Knoll's discovery down in Philadelphia. He actually was on faculty when I was in med school there. He got up there and lectured about it. We're all sitting there, wow. You know, it's really pretty amazing. And then we have monogenic diseases that we've defined down to the single chromosome. And we didn't need to wait for the genome to be elucidated to do that. So we had a relatively primitive means of starting to digitalize disease processes. And we're going to get that in a little while. We're going to be talking about sickle cell disease. So that's great. But of course, now we're getting these reams of genomic data from the patient populations, from normal populations, and disease populations. And using a kind of reference set, we see with people who have specific diseases, what lights up genetically. Sometimes it's a single gene. That makes it very easy. Oftentimes, it's a lot more complex, and using big data analysis and machine learning, we're finding relative patterns and relative risk scoring systems that seem to correlate with some of these imprecisely defined diseases. The good that comes from that is that it's that pathway towards a digital understanding of what's going on in pathology. The downside is when you reinforce some of the imprecision. You know, we're still looking for means of digitally defining things like autism, things like Alzheimer's disease, things where we don't really know the underlying mechanism, and we're chasing after associative pathologies like amyloid concentration in the brain. Now, it may turn out that that is the key that unlocks Alzheimer's. It may turn out, as we're finding with a lot of drug development in neuroscience, that in a lot of observations, it just doesn't seem to fit. And maybe it's just a detour. Now, polygenic, the problem with polygenic risk scoring is that it is mechanism agnostic in many cases. And without an ability to attach the mechanism to the phenotype of the disease, we can be misled by inappropriate biases in the sample that we're looking at. You know, one of the readings we had this week was the fact that we lack lots of diversity in what are turning out to be our reference genomes. So the polygenic risk scoring is very important for you guys to know the weaknesses of what are touted as some of the strongest tools that we have right now, because it's going to be your job to make them better. But at the same time, it's easy to hear about the more advanced way that we're applying them and assume that that's a standard, and assume that that's as good as it gets. And in many cases, these are examples of doing the wrong thing for the right reasons. The marriage of technology with biology is fraught with lots of problems. I see numerous examples of companies that are pitched to me that use the big data tools that have worked so well in meteorology, in crowd assessment, in taking a data set of observations of where the mouse goes on millions of people going to the same website and applying it to biology. And one of the observations I've had, again, just an observation, collection of analog anecdotes, has been many extremely high-functioning tech people consistently underestimate the complexity of biology. And that's one of the reasons I wanted to point that out in the reading. Other thing I wanted to point out was just the dangers of trying to glean data from unenriched samples. When you've got a group of people that all have the same symptomatology, and you go genetic fishing in there, you may find something that lights up in a much more specific and relevant way. But then you go out and say, okay, let's look for the same thing in the general population. And you may find that what was an acceptable amount of false positives and false negatives really leads you down the garden path towards doing the wrong things when you start assaying the general population. Third point I wanted to make just on this topic is, if you look historically, put them into a kind of knowledge perspective. The human genome was, elucidating the genome was an incredible feat. It was incredibly important. And we will be exploiting that finding for decades going on. It's going to be an enormous part of your careers if you stay in biology-based occupations. However, like every great new tool, we tend to overuse it and we tend to underemphasize everything else. So right now we're in a position where the genome means everything. The genome is immutable, it is the ultimate truth, and that means that we tend to underplay or ignore expression. We tend to underplay or ignore epigenetics. So it's important not to say, okay, the genome is everything. Our knowledge evolves. I've been trying to work on a convincing piece to write about gender, because it's such a political issue. And I'm a reproductive endocrinologist, and I saw many people come into the office that didn't fit into an easy XXXY binary. And you very quickly start thinking in terms of maleness and femaleness. And we need to update our knowledge base on this. It's much more complex. We lose the security of simple conceptualization, but on the same hand, we learn so much more. So simply counting the chromosomes, we invented a company that did the first embryo analysis, the first genetic analysis of embryos. And you just tested for XY, 13, 18, and 21 with fluorescent in situ hybridization. And you shine a light on the cell, and you count the colored dots. And this was around mid-90s to the early 2000s. It was terrific for some things, but it's super primitive medicine. And very quickly we realized that within the small biased sample set of having a XX or XY and two 13s, two 18s, and two 21s, there was a tremendous amount of other relevant diversity, obviously. That still holds true today when we're doing sequencing on just a few cells from an embryo. So it's really always vet the relevance of the data, the quality of the data you're getting, and never assume that just because it exploits the newest techniques and the newest tools, that it is, quote, state of the art. Keep trying to poke holes in it, because that's the way you're going to make things better. Look, there's a terrific podcast that I want you to listen to that talks about the evolution of CRISPR. CRISPR was invented in 2012. How much time has it had to evolve? It's a hell of a lot better now than it was then. 5 years from now, when I'm teaching this course, we're not going to be talking about CRISPR. We're going to be talking about something very different. So it's like, oh my God, it seems so precise. It is specific portions of the DNA. Cut them out, replace them. Well, frankly, we were saying the same thing 20 years ago about zinc finger transcription factors. And we were saying about something called talons. And it just didn't work well that well. They're still using it. So CRISPR itself is going to be relegated to temporary technology, the way all of this is. And it's happening very, very rapidly. So keep an eye out for that stuff. So the other message here is, when I send you this list of things to read and look at, do something. Okay, I'm trying to give you the foundation to build your own base for filling in what you don't know about biology, what you don't know about statistics. You know, the one or two of you who are economics majors, I really want you to learn a lot of biology here. And there's nothing we're assigning you that can't be understood and really brought in with just a high school science background. And I know you all have that. Okay? So, and then we went to the intro to venture capital. Very briefly on venture capital. We're going to talk about it explicitly later, but it's easy to kind of cliche venture capital. You know, if we do a word cloud for what you all think in terms of entrepreneurship, venture capital's there and it may be bold. And if I were to ask most of you to give me a precise definition of venture capital, most of you would not. If I were to ask you to give me a difference between venture capital and private equity investing, even some of you who have done one or the other in internships are probably missing that point. But venture capital and where it fits in a specific way is really a very important part of what we do. And we're going to have a little mini chapter on venture capital. But the seed I want to plant right now is I want you to start thinking for how we decide what the price is when someone goes out and buys a piece of a very, very, very early company. Remember, the biggest challenge, the reason biotechnology is entrepreneurship on steroids, is because of the length of the product development cycle, the sheer volume of dollars that are needed, and the specific amounts that always increase that are needed as you go along product development. So you need to have a parallel funding track that matches the needs of the program with each specific, where you are. So when you go from early preclinical, when you go from a petri dish to a mouse, to, God forbid, a primate, to humans, and you go from very small single dose to ascending doses to combinations to your efficacy trials to your proof of your regulatory meeting the needs of regulation, the costs just go astronomical. You need to be able to keep raising money. And in order to do that, you've got to have people with a very high risk tolerance. How do you attack that risk tolerance? You give them the basis to put a rational valuation on it, so that as I do this for a living, so that we can make rational decisions that the money we spend now is going to be on something that is highly likely to be worth a lot more later. Because if it's not, why buy it now? Why buy the piece of the company now? The challenge, when you go very early stage, is you don't want to, neither party wants to be exploited. You don't have enough information on which to base your handicapping of what the value will be at the next step. And the earlier you go, the wider the error bars on each of your estimation for how much the company's worth. So how do we kind of overcome that? We need to find a mechanism to make it easy to invest early without thinking that, boy, I'm stealing the company from the founders, therefore they're going to lose interest, and we'll have to rejigger this later on. Or, I'm paying way too much for this. So, just to give one of the tools we've used for that is we tend to try to avoid putting a hard valuation on the company in its seed in its earliest stages. So what we do is we use it's almost a convertible debt type vehicle, where I give the company a certain amount of money, we'll put the valuation on it later. And usually they say, okay, well, we'll put it up later. Eventually we've got to, decide what the value is. So typically the way these things work is you will invest in the company on a convertible basis, and the term that gets batted around most is SAFE. SAFE means simple agreement for future equity. And within certain parameters, we say, all right, I'm investing early, I'm taking maximal risk. On the same hand, I don't want to make an irrational decision on valuation, nor do I want the founder I don't want to bully the founder into doing the same thing. A, because being a bully is not a good way to go through life. B, Because you don't want to set up a disequilibrium later on when this same founder who you're investing in is again triaging how they're going to use the next few years of their life. They say, boy, I've already written this off. I've already given this company away. So I'm going to let somebody else come in and carry the ball, and I'm going to start something fresh. So you want to avoid that disequilibrium. So what you do is say, okay, let's push the decision down the road a bit and say that we will base the value now on how the value is defined later. So we do a seed round. The seed round is a convertible. We're going to wait a year, 18 months, 2 years, whatever the runway is. And we're going to say, at that point, we're going to have much more concrete proof of concept than we had before. We're going to base the valuation on that better proof of concept. And most likely, we're going to have another group of sophisticated investors joining us in this. Now you may say, well, why does that work for me now? Because I'm investing at a much higher risk point, and we may not attract that other group of investors. The experiments that we're basing on things may not work. So what you do is you negotiate a discount from what, for example, the Series A valuation. So Series A valuation is 50 million. You can say, okay, I'm going to get a 10, 15, 20, 25% discount in the amount that I buy, and what I'm buying down. Or I'm going to put a valuation cap on my investment now, so that you can say that, okay, the valuation is put at 50. I'm going to negotiate today that regardless of how the company is valued when you go out and you do your Series A, I will pay no more than the basis of the $30 million something like that. Okay? So it's a nice tool by which we can risk mitigate the unknown. And the risk mitigation is the key to making venture capital work. What we need to do is we need to let very sophisticated investors make rational decisions. If we were doing a private equity model, or if we were investing the way Warren Buffett and the value investors do, we simply plug in expected cash flows into a simple equation. We derive a discounted cash flow model. What's it worth now? Remember the lottery ticket. You can say you can get a big lump sum now, or you get $1,000 a month in your mailbox for the rest of your life. So you just find that equilibrium point where you are agnostic between one number versus the next. And if you're Warren Buffett, you say, fine, I'll buy the company for 25% less than that, or 50% less than that. And it's a simple equation. It's numbers, 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 easily, transparently, and reproducibly modeled. The challenge that the venture capitalists have is they don't have the luxury of working with things with such narrow error bars. In their cases, they're very, very wide. So the same concept of a margin of safety that you want to apply to decision making, you want to make in venture capital as well. Okay? Question, what's the difference between venture capital and angel investing?
Angel investing is normally like an individual or a group of individuals who are going to put their own money in, and they tend to not be as...
You got it. So stop there. Yeah, it used to be that people normally answer is, well, angel investors put in less. The only difference really is angel investors put in their own money. Venture capitalists, like we do, like I'm raising up a fund now. You go out, you get a group of investors to pool their money, and you invest for them. And angel investors invest their own cash.
They also invest pre-seed, right?
Angel investors can invest anywhere.
At any time in the process.
Yeah, it really comes down to, is it their own money or is it their investors' money? So you can have a syndicate consisting of angel investors and venture capitalists. So now actually we have some angels that are writing bigger checks than the venture capitalists, which is, it just is. Okay, one more question, then let's move on.
So when trying to avoid putting a hard valuation on something, how do you then determine what amount of starter money to do?
Well, we'll get into your decision for how much money you're going to invest later on. Simple question is, you say, okay, what do you need to get to the next level? You know, we're gonna talk in more detail, we touched upon it last week. Remember the 4 things that you're going after early on. One of the most important is proof of concept. And you're gonna have serial proofs of concept as you bring your company along. What's the first thing that you can go out and use? Proof of concept 1 may be your idea. And that's where you get your starter. And then, okay, how do you build the case that this is a business underneath that idea? In biotech, usually it's an experiment of some sort. If you're building restaurants, you may do a prototype. You know, if it's engineering, it's devices, it's definitely a prototype. If it's an app, you want to get the early functionality on the phone just to show it, and then back it up with data. Different proofs of concept. So that, you know, at that point, great, I'll invest at this stage. I love the idea, I love the founders, I love the way you put it together. It matches my own concept for where the world is going. All right, my money's gonna run out at some point. And I ask you, what's the next data point? What's the next proof of concept that we're building a business here? And you're gonna tell me that. And then I'm gonna say, how much money do you need to get past that? Because you don't want to be on fumes when you get it. You still want to deal from strength and negotiations. And that's the basis of our decision. How do we get to our next proof of concept? Okay, moving quickly on. Podcast I want you to listen to, seek this one out, is a guy named Harry Glorikian, who's a delightful, nerdy guy that I've known for years. He wrote a book called Moneyball Medicine, Moneyball based on the book about how Bill Bean for the Oakland A's digitalized baseball decisions. It's a great book, it's made into a great movie. Harry's book is great, he has a great podcast aimed at the digitalization of healthcare. And this particular one, he interviewed a guy named Kevin Davies, who's a journalist who writes about genetics. He wrote the book a few years ago, The $1,000 Genome, just wrote a book about CRISPR. He's a fabulous writer. I love learning from not textbooks because they're readable. You know, it's like you can say, okay, I'm done with my schoolwork. You can pick up one of these books and learn just as much. This hour or whatever where they talk, the arc of where genetics-based medicine goes is so beautifully demonstrated that if you're going out for a walk, stick your headphones on and listen to it. Very, very highly recommended. Okay, next thing I want to do briefly is I want to start setting the table for what's going to be an ongoing case study. And we're going to talk about the evolution of treatment for sickle cell disease and how it's responded and how it's evolved from kind of a systemic, symptomatic, reactive treatment that was pretty ineffective to one that's becoming increasingly focused, one that really lends itself to some of the tools that have been developed and that you guys are going to be developing over the next 20 years, and how we're going to be able to really change people's lives. Now, why sickle cell? For me, this one's personal. When I was, a million years ago, when I was practicing OBGYN, as a second-year resident running the high-risk pregnancy clinic, we would have a number of young women come in, 5, 6, 7 months pregnant, in sickle cell crisis. And we'll get into the pathophysiology of sickle cell disease at the time. But this was, you want to feel worthless, useless, and helpless. You have a woman in mid-pregnancy, writhing in pain, just having terrible, some of the worst pain you can ever experience. She's pregnant, so we're all concerned about the outcome of the pregnancy. All we could do was try to hydrate them with IV fluids and give them morphine for their pain. It was, you don't think you're a doctor, you just think you're a charlatan. It's amazing how humbling medical practice is. And sickle cell was just a perfect example of it. So, this is one of the things that my own personal interest is, my own entrepreneurial stuff, is we're trying to use embryo genetics to offer prevention to sickle cell for families that have had one heartbreaking outcome after another. As we get better and better with treatment, that type of thing may be necessary. But it's I wanna dig a little bit deep into the disease itself. So what is it? Most important thing to know for our discussion is it's very closely genetically tied. It's an autosomal recessive. It's a single point mutation on chromosome 11 for hemoglobin beta. So, the means by which we manufacture red cells. You end up with a mechanically flawed red blood cell. It's weak, and so it's like, imagine a mattress that folds easily, okay? And you've got red cells, little round mattresses that circulate through your bloodstream, and most of them have a good backbone, and they just manage to, they're flexible when they need to be flexible, but they're not too flexible. Sickle cell hemoglobin results in red cells that fold. Under certain conditions, whether it's dehydration, whether it's inflammation, you trigger a folding of the red cells aggregate, they don't travel the branches of the vascular system as easily as they could. They get stuck, they form clots, they form infections, they cause organ dysfunction, and the symptoms are periodic. So what you have is people will appear in very acute pain, or they will appear with very acute infections. Over time, there's organ damage. The organ can be the liver, it can be the spleen. These people often have to have their spleens removed. It's just a series of awful things they have to put up with. And over the years, from when I entered medicine to now, we've been able to exploit some of our discoveries to make better and better treatments for it. And we're going to kind of walk through that. Is it serious? It's damn serious. Symptoms start very, very early in life, and the childhood mortality is unacceptably high. And so, we talk about entrepreneurship really above all being the solving of problems. This is a hell of a problem to solve. So you see something like this, you say, okay, just stop there. Let's just go to work on this. So who's affected? You see I'm building my pitch here, and I'm constructing my numbers. It predominantly affects African Americans or people of African descent. It's not only Americans, we tend to be very US centric. And from certain Hispanic countries, we're at the World Health Organization, about 1,750 cases per year in the United States. And let's back that out to a different number. That means close to 8,000 pregnancies at risk per year. Okay, everyone knows the difference between incidence and prevalence. Prevalence is just the number of people affected by something at any given time throughout the population. Incidence is the new cases that have come up per year. So 1,757 cases, about 8,000 couples trying to have children or having children without trying, without planning. How many people are born per year in the US? And we'll always have these big numbers in the back of your mind. A good number to think of is 4 million. It's actually about 3.75 million right now. There's concerns about population shrinkage. But if you have a big, approximate big number, 4 million. So it's a relatively small number per all the deliveries we have per year. But you divide that by the number of hospitals, number of cities, it's not that difficult to see that anyone in a busy practice is going to encounter people with sickle cell disease. So it's important to get first. And overall, there's about 100,000 people suffering from it now in the United States. Management options over time, first we're just symptomatic. It comes from the crises come with dehydration, with dehydrate. Stick an IV in, just pour fluid. And you manage the pain as best you can. You treat the infections when you can. You prevent sepsis, which is if you're going from a localized infection to a systemic infection in blood with a high fatality. You manage the chronic problems as they come up. You may need to remove the spleen, for example. You may need to treat renal damage. So there's a lot of things going on. So we've gone from there. The first drug that's been used sort of effectively is something called hydroxyurea, which really just makes the cells a little bit more slippery. Again, we're going after the end pathophysiology. This is not a cure. This is a means of facilitating getting rid of the symptoms. Now, what do we do for disease modifiers? How can we kind of prevent the disease at its source? And we'll talk about that over time. More importantly, how do we go after cures? It's a genetic disease. We now have genetic tools. And we're going to talk in some coming weeks about how we can use those different genetic tools to maybe cure the disease. You know, the mythical, magical one and done treatment that comes in, medicine like surgery. You remove the disease process, and the person goes on and lives a normal life. And as I mentioned, my own interest is in how do we prevent it altogether. So sickle cell is a great platform to which we can attach some of the things we've been talking about over the evolution of biotech right now. Okay? Let's push on. Okay, a little review of what we've done so far. Remember the first week, we went through the rules for people. We wanted people to walk away if they weren't going to take the class, please. Next week we went to, you decide you're an entrepreneur, what do you start doing? Chip, can you see the slides? Okay, I gotta make it so that everybody can see you. I'll get that up in a minute. But, so you decide to become an entrepreneur. Remember, we talked about 4 tracks that you follow. There's the build an organization, learn how to communicate track. There's the seed capital, the funding plan. Define what's proprietary and protecting it, and very importantly, establish proof of concept. So this is your world from day 2 of being an entrepreneur. Okay? The 4 different things you keep running through in your mind. How am I paying for all this? How am I showing it's a business that works? How am I building a legally acceptable organization and knowing how to talk about it so that I can attract colleagues and coworkers and funders and BD people and things of that sort. And finally, figuring out what do you do for protecting the company? What is proprietary, if anything? And what do you do to protect it? Now everybody thinks patents, patents, patents. There's a lot of different ways of approaching it other than patents. The thing is, we'll focus on patents mainly because people don't know the real specifics of them. And patent strategy is very important. So, you go through the branch of identity, putting together your identity. Remember this flow chart from before. Give it a name, give it a description. Get involved in conversations where you let the world know you're building a company. And you wait for tell me about it. A lot of people are going to want to say tell me about it. You give them a description, tell me more, then you go into your elevator pitch. How do I follow up your business plan? Okay? Within the context of all the creativity that entrepreneurship offers you, it's good to have a scaffolding of a script to base it on. Make it your own, bring your own personality, your own originality to it. But this is a way to very rapidly fulfill that obligation you have, which is to separate the world into the people that care about your entrepreneurial venture. They are on the partner, employer, funder, business development person, mentor, board member track. And everybody else. They may be on the friend, potential spouse, someone you'll hire as a nanny, dog walker, person you go to concerts or baseball games with, anything. A lot of roles for a lot of people in life. Your goal here is, early stage company, gather in, enrich the population of people that are going to help you out in the company. So learn them quickly. Separate the world that way. Now, it doesn't mean people can't jump from one group to the other, but it's a good idea just to limit your discussion of your pitch and business plan to the people that care. If you run into your elevator pitch and everyone you encounter, you're going to get really frustrated about a number of people that you really don't want to listen to. And on the same hand, you're gonna waste a lot of time. And this is time that could be making friends or going to see Yankees, whatever like that. But on the same hand, some of these conversations get very long and very detailed. And you want to protect your time so you can apply it to those people. And a lot of this is informal. A lot of this does not come from this business development conference or that business development conference. I hate the concept of paying to pitch. You will be getting asked a lot to pay to pitch. Oh, we're putting together this forum, that forum. We're bringing in 1,000 VCs to listen to a series of pitches. I've gone to some of those. They are relatively low yield. You are more likely to, in the serpentine Brownian motion method of network, find people who care, one at a time, than going to a vast stadium of potential investors. It kind of goes back to the concept of testing the unenriched population for genetic trade. It's like you bring in people that can do, they might be there, they want to invest in gun companies or something. You don't know. They're not, the people that organize these meetings are not necessarily that focused on what you're doing. That said, there are tremendous opportunities to meet people who care when you go to aggregations focused on the subject matter. You go to Chip is on one of the directors of Mass Bio. You go to a Mass Bio meeting and talk about your company. You don't go in with the expectation you'd be pitching for investors, but you're likely to find people that want to invest in the types of things you do. If you stick to, the closer you get to what it is that you do, you may find it's, first of all, you have to pay to do it. It's more interesting because you're talking about people with a similar vocabulary. And they're often, anyone you talk to, regardless of what their role is, they may be 1 degree of separation away from the person you really do need to talk to, okay? So that's a kind of a review of what we've done so far. Okay, I'm not going to call people out by name. I am calling you unis, and I want you to elevator pitch either what you used to get into the class for the email you sent with your idea, or something that you prepared since you got the assignment to be ready to pitch, or without giving it away, something you were making up on the fly because you walked into class woefully unprepared. Okay? Any of those is acceptable. So, boy, is the type small on this sheet. SMC 2304. You do it from there, or you come up in front, whatever you want. Just, I realize you're wearing a mask, so you have to project.
Will do.
Okay. You can use your name if you like.
Alright, so hi everyone, my name is Salvatore Caruso, I'm a second year PhD student.
Come a little closer to the minutes.
Is it my
Okay.
So I guess the working title of what I'm presenting to you guys on is called Precision, and in 6 words, it's gene editing for inherited retinal diseases. So I work in Dr. Sang's lab up on the medical campus, and we specifically focus on research and rare inherited retinal diseases. And so my platform essentially uses cutting-edge gene editing technologies like prime editing to essentially correct mutations for RDS-linked retinitis pigmentosa. For those of you who don't know, it's blindness-causing disease with approximately 4,000 annual cases every year in the US. So it's relatively small. But when you look at what's already been approved for FDA clinical trials, you see that drugs like Luxturna, which has an RP65 linked retinitis pigmentosa approved.
Okay, I'm going to stop you there. Okay, great job defining what you do. Good job defining why we care about you individually doing it. You haven't really told us what it is that's magic about it. You got to get there closer. Okay? Here's the problem you're solving. Okay? Here's who it affects. You threw a number in there. It was a little vague. We still don't know what it is about what you're doing. Okay? Don't lead with the idea that that's 2 bullet points down, you got to hit. So, describe succinctly what's different about what you do.
It is a prime editing platform specifically for the correction of RDS-linked retinitis pigmentosa.
Okay. Great. Excellent. Okay. They're not doing very good. SEC 222.8 for 6. We need a better printer.
Hi everyone, I'm Sarah.
Louder.
Not loud enough. I'm Sarah. So essentially, my technology is based upon type 1 diabetes management. Currently, the technologies that exist are targeting hyperglycemia. So what that means is it's high blood sugar. So management technologies include insulin pumps combined with continuous glucose sensing, and these are great technologies currently. The only problem is that it's invasive and it's not really talking about the opposite side of it, which is hypoglycemia, which is low blood sugar. So if you take too much insulin, it's going to make you go low. If you exercise too much, it's going to make you go low. So you have to think about all of these things. So essentially what I want to do is combine insulin pump with not only just insulin, but also glucose giving mechanisms like maybe glucagon or just straight sugar. It's kind of nice to have that all under one roof, I think, because especially when you're not, say you're going for a walk or a run and you forget to carry glucose or something with you, then you already have it in your pump and it just automatically would deliver it.
Sarah, I'm going to stop you there. Who are you pitching to right now?
I guess diabetics.
Okay, so as much as we want to keep the pitch uniform, you got the 5 things you want to hit, okay? Problem you're solving, who it affects, what your idea is, why you, and then some number to give us an idea of what kind of business is underlying. You got to kind of keep a consistency to who you're talking to. If you're talking to a group of upper level undergrads, early grad students at Columbia, all of whom got a basis in science, that's good. If you're talking to a patient group, that's fine. If you're talking to a extremely loving older relative, that's good too. And if you're talking to the most human-hating, awful end-of-career venture capitalist who's hungover and in a bad mood, that's something else. You're doing something we all do, is you're jumping from target to target, okay? So everything you say is great. You didn't include the name of the company, which I always, remember, first meeting, we want to make sure this is a company, not your hobby. So lead with the name, okay? It's just one micro validation that kind of makes us take you a little more seriously. The much more important validation comes from vetting your presentation, your knowledge, and then remember there's the 2 types of questions we ask. There's the question to better clarify what you're telling me, and then there's the question to try to get an idea for how you work. How deep is your knowledge? How strong are these numbers? What's the likelihood of you making something up? Okay? So, all good. But we could be a little bit too democratized in the way we present. We want the loving great aunt to understand, and at the same time we want the senior, the PI, the person that does the scientific vetting for the multi-billion-dollar VC fund to understand as well. So work on consistency. Not that, now I didn't tell you who you were presenting to, but it was a perfect example of that. Very good. Okay. All right. Oh, not only is the type small, but it's smudged a little bit.
Yeah.
JT 2411. JT 2411. G. Taylor. JT 2411 is in. Yeah. Come on up and pitch this. You can do it from there, you can do it from here, however you like.
So I think I'm not ready yet.
Okay, I'm sorry, I can't hear you.
Sorry, I think I'm not ready yet.
Okay, have a seat. AP 4145.
Oh, that's me. Okay.
I
Okay,
know,
so,
we're not used to be calling out our unions.
Yeah.
But
So
if you all as a consensus, don't mind my using your names, but, you know, we're recording these and things of that sort, so I wanna give you the opportunity for anonymity if you want, so. We will have masks on too. Okay, go ahead.
I
Pitch something
don't
to
have a
us.
company name yet, but the problem I'm trying to address is that
Little louder.
in underdeveloped communities, a lot of people, especially women, don't know about the repercussions of not protecting your reproductive health. So I'm trying to come up with an at-home testing apparatus to test for like STDs, STIs, in the event that a gynecologist is far away, if they're in like rural communities, and so everyone can take care of their reproductive health.
Okay. Start again pitching to your great aunt.
Oh God, okay. Okay, so my great aunt is from Nepal, and that is an underdeveloped community. And so a gynecologist's office would be really far away. And so I tell her that,
I am her. I know.
Oh, okay.
I look
So I want to make sure your reproductive health and those of your friends and your family members is good and healthy, and I want to make sure that you have an at-home testing apparatus to test for your reproductive health, and because that can lead to high-risk pregnancies or other health issues down the line. So in order to make that more accessible and affordable, I want you to have an at-home testing apparatus.
Okay. Yeah. When I had them, my great aunt was much older or a different era. If I used terms like high risk, at-home testing, reproductive health, she would look at me with her loving eyes and have no idea what I was talking about. So now, it's likely that your great aunt is much more sophisticated that way than mine was. But we take for granted the level of understanding certain people have of certain terms. Particularly, you go from the US to Nepal,
Yeah.
and there's different dialects that we use, particularly with a health vocabulary. So it's just something to be aware of. And it's something more of you aware of, just kind of sense how much is getting through to who you're talking to. You may find that when you shift gears, this is something you never taught us in medical school, but somehow we all learned it. You know, I found that there was a subpopulation of my own patients in OBGYN where if I described a body system and compared it to the working of an automobile, everybody got it. It's probably has something to do with the fact I practiced in New Jersey, which is just one big pile. But you will develop a different set of vocabularies, as well as just kind of this sensor, this Bluetooth from you to your listener that tells you what's landing and what's not. So now, stand up again. I don't mean to really put you on the spot. Remember that awful venture capitalist we talked about before? Okay, that one looks a lot more like me. Why don't you pitch your idea again to someone like that?
Okay, I wasn't here for the first class, so I don't know.
Okay.
Okay.
Just someone with a highly technical knowledge, someone who's sort of impatient, sort of, not the nicest person in the world. Someone who may look at their watch while you're talking, who may look impatient, but for whatever reason, they're sitting across the table from you and you're talking to them.
I wouldn't know where to start.
That's the perfect answer.
Okay.
The point I'm trying to make, and we're gonna flesh out, you can sit down. We're gonna flesh out how to approach these kind of situations. The number one thing you need to sit down and think about, if you're talking to someone who's really intimidating, who you think has a disproportionate power relationship in the whole thing, is you sit there and say, I'm okay, you're okay. I know what I'm doing. It may not be right for you. You're thinking this, you're not saying it. It may not be right for you. I unashamedly own what I'm doing. If it's not right for you, fine. That's okay. You know, we may cross paths 10 years from now when I'm doing something that you really care about. Or I'll never see you again. You know, don't let the scenario itself throw you. You know, there's particularly for founders, particularly for female founders, particularly for founders of unrepresented groups, there is some really disproportionate power relationships that you'll find yourselves in. And just shrug them off. Remember, rule number 1, be the calmest person in the room. Be the calmest person in the room there. You will find that not only will it make them tolerable to pleasant, it's validating. You know, the person that comes in all blustery like any other potential bully, if you just like, that stuff doesn't work with me. This is what I'm working for. You know, if you want to end the meeting early, just end it. It's like, you know, this doesn't seem like this is really, you know, something that's in your sweet spot. You know, listen, nice meeting you, something like that. You may find say, no. You know, as soon as you have given off that you're not desperate to keep their attention, suddenly you become much more interesting for them to talk to. So this is not going to be the majority of your meetings, but you will have them occasionally. First thing, just set the parameters. I'm okay, I'm supposed to be here, I'm doing something really valid and great. I assume you are, you're certainly acting like you think it is. And just take it from there. You are on a level playing field if you choose to be on one. So, you know, there's never, you know, we're going to get into when we talk about negotiation, a similar thing. You always want to negotiate from strength. The single most important thing that helps you negotiate from strength is being able to get up and walk away. I'd love to do business with you, I don't have to do business with you. Okay? A similar dynamic occurs during the pitching process. And whether it's regardless of who you're pitching to, or in the hiring process, whether you're hiring or being hired. Okay? So you want to enter all of these interactions. And it's not intuitive in your stage. You know, you're thinking, okay, I'm a junior person, just out of school. You know, it's like you see, I don't know what's so intimidating about gray hair, but somehow people take me a lot more seriously for no good reason now than they did when I was your age. I think I was a lot more interesting when I was your age. But, so don't let the scenario, don't let someone's title, don't let someone's bearing throw you off, okay? You don't have to be there, you don't need them. And as soon as you set that tone, most people will rise right to it. They will say, oh, those are the rules, great. Okay, yeah, you've established them. That is so validating from the beginning, okay? Got 2 more minutes. I do not want to take 1 minute away from Chip. Any questions? Question?
Yeah, I have one question. So, like, some people have the benefit of having like a really common disease that they're trying to cure, so they don't
Yes.
need to go into the science at all. And I feel like for me, like, at least when I was up there, I just assumed no one knows anything about rare retinal diseases.
No,
So
it's
how do we do that in like a time concise manner? That's where I lose a lot of time on my pitches.
yeah, you know, is it a cause of blindness? At what age? Okay, how rapidly does it happen? And it's a incurable, rapidly progressing cause of blindness that affects people this age. That's about 20, at most, 20 seconds. You've got their attention. If they know this, you're not giving them too basic if they even bore them. If they don't know it, you've given a really good, concise perspective by which to assess everything else you're saying. 2 more questions and we're done.
Just wanted to highlight on that. Doesn't that reiterate the point that you want to pitch to VCs that operate in your vertical in any case? Then they already know what you're talking about.
Yeah, that's great if you can. You know, and you know your audience. Now, there's a feeling out process. Like when I don't tell people that I practiced medicine for 12 years. I don't tell people, you know, like boards and stuff like that. if they start going into a little too much detail, I'm saying, yeah, you can skip this. Let's go to, I like to see the slide deck in advance. I'll say, let's get to number 17. You know, I'm good on the background. And that's good, they appreciate that too. One more question. Yeah, okay. Let's, now, I am thrilled that Chip, I gotta make this work. Can you hear us? You're on mute. Okay.
They were standing in front of the class, but I have not consistently heard the dialogue from the bleachers.
Okay, so when questions come, loud. Okay? Now, can you see the class? I don't think you can see the class.
I can't.
Let me see if I can find a way,
There we go.
because you want to be able to know who's out there. Okay. Now, class, meet Chip. Chip, meet the class. Chip, I do this the same way every time. I gotta see if somebody can see me. Here. Am I on something?
Yeah.
Okay. One question we're gonna lead with, which is, to look at you, it looks like you were sitting in a class like this very recently. Now, could you, in whatever way you want to tell your story, tell us how you got from there to where you are now? The students know your background, know what you do now, but in any level of detail, focus on whatever you think is important. My job is to make sure that they hear your story, and I'm gonna give the floor to you.
Thank you for the invitation. I really enjoyed listening into the class.
Can you guys hear?
The intersection of medicine and business is fascinating and challenging and exciting. And so hearing the way you're teaching it is I think enlightening. I've also enjoyed hearing the pitches. They're quite cool. And I'll say to I think you're sitting in the front there, the Precisio founder, hopefully founder. Retinitis pigmentosa runs rampant through my family, so you never know when you're going to run into people who do actually know in multiple layers of detail what it is you're pitching. But as David rightly said, have multiple layers of pitches ready to go depending on your audience. If it looks like I've been sitting in your seat recently, then the gauzy settings of Zoom that I deliberately set are working very well. I have sat in your seat, and maybe I'll tell you who I am and then sort of how I got there. So I am the CEO of a company called Genocea. It's a public biotech company based in Cambridge, Massachusetts. We are developing personalized cancer immunotherapies for patients suffering from polygenic cancers, meaning cancers that are not arising from one specific and well-known mutation. Our solution to that problem is to find ways to harness the patient's own immune system to tell us the relevant cancer targets and to focus the therapies that way. It's in some ways, I think, a really interesting approach to the problem that David was laying out earlier. This idea that machine learning and new analytical tools are incredibly exciting about their potential, but may in fact have limitations in the degree of certainty they can provide us as we make therapies. Our approach, of course, has its own pluses and minuses, but it certainly is kind of built with the understanding that algorithms and machines lie sometimes, and that we had to find ways to get greater certainty in our targets. Maybe to tell you just how I got there, I was an economics major in college, but I was also really interested in science and medicine. I took several college level science classes, and even though I stayed an economics major, I probably was really fascinated by the science because it frankly gave there was sort of a right answer, and it was clearly defined in many cases, which maybe sounds ironic given all of the uncertainties associated with diagnosis and treatment today, but usually there was a correct answer, and to some extent I found that reassuring in a world where economics to this day continues to struggle to find ways to point us unerringly toward the future. But I went from an undergrad economics degree and jumped straight into the pharmaceutical industry because I was fascinated by the science classes, and because I, in some ways, was maybe a product of my times. This was the late 80s and early 90s when, believe it or not, Merck was voted the most admired company in America. There was a time when pharmaceutical companies were the most admired companies in America, and I joined another company called SmithKline which is now part of another mega company, GlaxoSmithKline. And during my time there, I got, I think, a really nice grounding in the basics of drug discovery, development, and commercialization, the overall business through a couple of different roles in the company, starting from pharmaceutical sales and working through R&D project management and strategic planning. So great sort of understanding of the fundamentals of the business. I then got an MBA, and then, not at Columbia, unfortunately. I went from there to biotech venture capital, which was, in a way, a great reintroduction to pharma, because I had been coming at the value chain from the perspective of the largest companies with seemingly infinite reach and seemingly infinite resources. But, at the heart of small biotechs is, as David said, it's solving an unmet problem. And I think bigger companies have, in some ways, the luxury of resting on their laurels and kind of milking every last dollar they can, frankly, out of treating as many patients as possible, or ensuring as many patients as possible are treated with their approved medicines. But fundamentally, you have to have a great idea, you have to advance it in the most thoughtful and capital efficient way, and then by doing so, make it available to as many patients as possible, either by yourself or through a big pharma company. And what I learned as a biotech venture capital was really how to translate that sort of idea or molecule to finish line model as if it were a small company. And so I had the great fortune to look at hundreds of companies that were just starting or were in the middle of their path from the beginning to commercialization. And it really helped me solidify in my mind, first, that I really wanted to do that, rather than kind of be the one just giving money to the people who were doing that. And secondly, it probably helped reinforce in my mind the strengths I might have in bringing such company or companies forward, as well as the things I need to continue to work on. So while I was a venture capitalist, I worked on a business plan for and ultimately launched a company from that firm called Vanda Pharmaceuticals. The idea behind this company was that pharma companies were in the midst of a business model crisis, were paring their development pipelines, were discarding potentially lots of good drugs. Our goal was to rescue good ones and focus them where they needed to be. And our specific focus was in CNS diseases. First indication was a schizophrenia drug. And so took that company from business plan through a couple of rounds of venture financing, of course, acquiring assets, building a team, advancing the lead compounds, going public, getting the first drug approved, getting it partnered with a big pharma company, and so that makes the story sound entirely linear, which it was very much not, but it was an incredible 7 years of my time, it catapulted me to Genocea, which I guess I say is my second startup, but in some ways could be considered my third. As David may remember, since we met many years ago for the first time, Genocea used to be an infectious disease company, and built around the same principle that algorithms can't tell us the right targets of, in this case, responses to T cells, for T cells rather. And we had a company that was, I think, scientifically and clinically successful in many ways, but ultimately failed to compellingly prove that we had solved a real patient problem. We were, to be more specific, developing a therapeutic vaccine to treat genital herpes infections. And we were successful in multiple clinical trials demonstrating positive data on, excuse me, the endpoints of success, the impact on disease as measured in the clinical trial, and measures that matter to the patient's insurance, things like that. But sort of the magnitude of the improvements, just based on the fact that we were unable to sell the story in a compelling enough way to either the potential pharma companies who could advance the program, or the investors who could help us do so instead. You know, we were faced in sort of late 2017, early 2018 with a sort of quit or reform question. And we ultimately decided that our technology was sound and that the cancer immunotherapy revolution was real, and that our platform could potentially solve this problem of how to direct vaccines and cell therapies to the right cancer targets. And so we undertook some wrenching decisions to lay off most of the team. We went from 100 or so people to about 30, to find a whole new set of investors around a completely new story. And that's what we've been doing over the past few years, and we hope, on the precipice of really more conclusively proving that this is the right decision. We're in the midst of a clinical trial now, and expect to have the initial data from this sometime in the next, sort of half year or so. So that's me and sort of a quick version of the story.
Questions? Hey, Chip. I've got one. I'm trying to figure out where to look. You've done big pharma, you've done startup, and you've been in venture capital. One of the things we stress here is that you can do the problem-solving aspect of entrepreneurship in almost any setting. The one that seems least intuitive is doing it within the context of big pharma. And you alluded to that before. Could you expand on that? How did that come about? Was it what you expected? And how did you find that you were able to be an effective, essentially founder role within a big pharma company?
Yeah, no, thanks, David. I think there are a couple of challenges that big pharma and probably all large companies face. One is to move with agility and speed. I think that large companies necessarily must bring together large groups of people and kind of move in unified directions, and sort of getting there, whether you're talking about decisions about molecules to prioritize in the drug discovery to whether you're talking about sort of commercial strategy for your approved products. Just decision making just takes so much time. It's the proverbial aircraft carrier that you're trying to steer, and there are lots of people with jet skis zipping around the aircraft carrier perhaps not moving with as much power, but much more nimbly handling decision making and course correcting. So I think that's one potential challenge. And I think pharma companies are trying to recognize this. The company I came from, GNCA, ultimately decided to break their R&D organization into what they were, I forget the name they were using, but it was essentially sort of mini biotechs focused around different mechanisms of action and or therapeutic areas. And they weren't able to make it work, probably because of a lack of commitment, a lack of patience, and for sort of mistakes and sort of timeline slippage. But I'm speculating here. So I think that's one potential challenge. I think another potential challenge is just diffusion of responsibility. Who owns the decision making in such large companies? During my time at SmithKline Beecham, there were, I saw two, extraordinary CEOs. Extraordinary in the sense that they were able to have their fingers in nearly every decision of consequence and do so in a thoughtful way. But the consequences of doing so in a 50,000 person company are that everybody's looking upward. Everybody is not trying to do what they think is right. They're trying to fit within a process that feeds this beast, this person at the top of the organization who probably ended up making good decisions much of the time, but by putting themselves in such a position, absolutely, I would say, demotivated many other people, and had us thinking about not for a given molecule or clinical trial or campaign, what is the right thing, but it's how do we get him to tell us what the right thing is. And that's not exciting. I think what I found from being a VC, as I said, was just the idea of having a more impact personally, more direct control over the fate of the company, and having a clear ability to not just solve problems, but also to think holistically about what we're trying to do in terms of solving the big picture problem of meeting an unmet patient need in a way that's going to be compelling for patients, for providers, for payers, in a way that I think it's just really challenging for big pharma companies to do. And I think that's one of the many reasons why our sort of biotech ecosystem has thrived. Plenty of individual companies fail, but others succeed. And if you think of the clusters of biotech companies in Cambridge and San Francisco and San Diego and Research Triangle and all of these other places, and that's a very US-centric answer, I will admit. If you look at the productivity we've demonstrated over the past 20 years, I would be willing to bet that somebody like Bruce Booth has done this analysis. I bet that we have developed more compelling drugs in a more capital efficient way, ultimately, than the pharma industry has.
Questions? Questions about it?
Hi, thank you so much for coming.
I'm not sure you can hear me.
Can you hear me okay? Okay, great. When your company was originally tailored for infectious diseases, you said you had an issue, proving to your stakeholders. I don't think you really had an issue, but you had an issue proving to your stakeholders solving the problem. If you could go back in time, what do you think you could have done differently to maybe keep them, invested?
That's a fantastic question. Thank you. It's a question I spent many sleepless nights thinking about before ultimately moving on. But I would say that in some ways we were too conservative, and one of the challenges I think small companies always have to ask is how much risk to take. There's of course risk in starting a company, working as a small team, when, at least conceptually, you might be fighting against larger pharma companies or major university labs or other things. And so, one of I think the job of a CEO, the job of the investors and the board of the company is to make sure that the company is allocating capital in the best possible way. And I think in this specific case, the thing I would have done differently is run a higher risk trial. And what I mean more specifically is this. In the case of genital herpes infections, there is a pretty good approved drug to treat the infections that is pretty good in the sense that it's pretty safe. It does a nice job of knocking down the symptoms and transmission risk of the disease, especially if you take it every day. And when we were developing this vaccine, we were, in our minds, positioning it alongside that therapy. And we convinced ourselves, and in my opinion, we had data in our hand that said that just taking this vaccine once a year after the initial couple of shots would be just as good as for patients as taking a pill every day to treat the infection. And I think we succeeded there, but I think had we shown that instead of offering something pretty good, that in fact there was a solution that, let's for the sake of argument say, essentially eliminated the disease. We weren't going to actually clear the virus from the body, but if we provided, very reasonable assurances, 98%, 99% assurance that the patients could have a comfortable life. I mean, genital herpes doesn't kill you, but it affects you in a very human way, the way you are a sexual being. And that's core to so many of us, almost everyone. If we could have provided greater assurance there, I would bet that we would have been able to succeed. So we were not, I would say, bold enough in our clinical strategy. We opted for the trial we were sure we could win with clinically, even where the probability of succeeding commercially was lower. And we probably should have, I would say, taken more clinical risk, because had we succeeded there, the commercial payoff would have been more certain. Does that answer your question?
Yes, very much. Thank you.
No, that's a great question, thank you.
Zoe, your question.
Oh, can you see me?
She's in the corner.
Yeah, sorry. So the company I'm currently working at focuses on CNS and
Oh, she's
stand up.
just so they can see. There we go.
Hello. The company I currently work for focuses on CNS and oncology, and we're actually doing a large consortium with JANDA right now. So I'm wondering how you found the transition from CNS to oncotherapeutics.
I think that's a great question. I found it very hard, to be candid. Not that any disease, not that, well, let me take a step back. I think in order to be effective as a leader of a company, you have to sort of generally know where you can have impact. And I think, and to focus your efforts there. And in my opinion, that means certainly understanding the science of a disease, the clinical practice of the disease, and from those things, where the unmet needs are. And then in the context of a company, thinking about whether it's drug discovery, the development, the clinical trial design, the partnering efforts, the commercial planning, the selling of the story, which are of those sort of aspects of the company where I can, play the most impactful role. I think the biggest challenge, honestly, I found in the transition from Vanda to Genocea, beyond the team, because missing the Vanda team was like a family, was on the science. Cancer, in no way to sort of denigrate the science behind the treatment, for example, of schizophrenia, which is a tremendously complex disease, but cancer and cancer immunology is, in my now reasonably informed judgment, enormously more complex and enormously more complicated from a strategic perspective as well. You likely know that psychiatric diseases are still today not given enough attention. And that's in part because of still today a sort of a nascent understanding of how the brain works. And so from that, there's many great avenues to advance new medicines. But there's been an incredible explosion of knowledge in the mechanisms of and the development of cancer. And as a consequence of that, whereas at Vanda, we were thinking about 10 companies as sort of peers to watch. In cancer, it's hundreds, and the number seems to grow and change every day. And so not just thinking about the tremendously complex science of what we're trying to do, but also understanding how, because our job is to fund and continue to sell our story, understanding how to continue to think about the evolution of the broader understanding of cancer and where our therapies may fit is a challenge in and of itself. And so, and from that comes another big challenge, which is sort of getting attention. When we had a novel schizophrenia treatment, that was not particularly hard. People sort of grasped that schizophrenia is a terrible disease and that there weren't that many sort of good treatments for it. Whereas if there are hundreds of companies with novel cancer therapies, telling a story, and we're practicing these things earlier in the class, telling a story in a way that, and then really constructing a story and telling it in a way that commands the attention of, for example, investors, for example, the potential larger biopharma partners, is something that we have to do with great vigilance, because what worked even 6 months ago has to continue to change in response to the evolving times. So whereas in this case with Vanda, we sort of set a plan and more or less executed on it for 3 or 4 years with Biotechnology and that. Our cancer plans have evolved substantially even in the past couple of years. So it's a great challenge. It's a great problem to have. It definitely keeps us on our toes, but it's a significant challenge.
Thank you very much.
No, thank you. It's a great question.
Hi. So you've dealt with a lot of investors, and I was kind of wondering if you've ever come across an investor who was interested, but for some other reason you didn't want to work with them. And I was kind of wondering how you pick and choose it's more like how you pick and choose which investors you want to continue to work with and who might not necessarily, whatever your experience is with that.
Yeah, no, great question. So yes, I have encountered such investors, and maybe I'll distinguish investors between ones that may, we're a public company, as I explained. But let's go back and think about how we got there, which is through venture funding first. And I think the critical questions that we focused on related to your question was, which are the right venture capitalists to work with, and which ones did we want to be advisors and board members, real partners in the development of the company, versus ones who provided money, and which is the lifeblood of the industry in some ways. But that's mostly what I wanted from them. And so I'll tell you the answers that worked best for me. You know, when we were constructing our board as a private company, we were trying to position Genocea in a way that we thought would be most compelling. The problems we wanted to solve, and therefore the things that we were most interested in from potential investors were, probably in no particular order, we wanted branding. Again, so you want to kind of be able to stand out, and so you're looking for venture capitalists with an established track record, and ideally with a relationship with the partner who has a track record of success and perhaps is a name. The name provides a little bit of flash value. Goes on a website, that's good. But we were also looking for people who brought specific experiences to the board that we may have lacked. What that means specifically depends on the company, of course. In our case, when I took the helm of Genocea, we had a smart team, but it was not a particularly experienced team in managing a portfolio of drug candidates. There are people with lots of experience in managing one, in other words. And so we were looking for venture capitalists with a proven track record in helping companies who were wrestling with exactly that kind of problem. We certainly were interested in their specific technical expertise. In other words, a familiarity with cancer and cancer drug development. Well, actually, in this case, it was infectious disease at the time. With infectious disease development was important, and that's in that most, or probably at least the majority of VCs have MDs or PhDs. That wasn't too much of a problem. And I think all of those things mattered, but none of that would be useful if, I didn't like working with this person, and or other board members didn't like working with the person. That really matters, not just because life is too short to work with people you don't like, but because if you like someone and they like you, and I don't mean that you're necessarily friends, but there is a collegiality that brings the best out of you, and hopefully you bring the best out of them, is so important. And it's really important for board dynamics as well, because every time a venture-backed company board meets, in my opinion, it should be just focused on one or two strategic questions, which are usually variations on the theme of how are we going to get the next slug of money? What does our story need to be to get there? And how are we tracking internally and strategically in our plans to do so? If the board dynamics are not good, those are going to be terrible meetings, ultimately unproductive. So those were the things that I was thinking about, that I continue to think about as I now construct my board too, which has less to do with investors and more to do with other sort of capabilities. But yeah, I absolutely encountered investors who I didn't sense I would enjoy working with, or I didn't sense would fit my board. And I think the best thing I could do for myself and the company was to have options. Create as much optionality as I'm sure all of you in this classroom have gotten to where you are by embracing that mantra in some way, shape, or form. Create as many options for yourself as possible. And so we certainly kissed a lot of frogs, so to speak, to find the investors that we most wanted to work with. Did that answer your question?
Yeah, it did, thank you.
Right next to
Hi, it seems like a lot of the solutions to some of these drugs that get abandoned during development or these failures tends to be changing how the story is told and how to really justify solving that problem. How do you kind of balance giving a really compelling story without over-promising or being dishonest in order to get a really great idea out there helping people in a way that really appeals to investors instead of kind of like your story with the infectious diseases?
Yeah, it's a great question. I'll borrow a metaphor that I learned from one of my board members who I know David knows, Katrine Bosley. And she says, you have to have sizzle and you have to have steak. And I think the balance to strike in telling a story is all about having both in the right mix. And what I mean is this. I was listening to the pitches that we heard at the end of the class, and I was struck by the second one. In that, and this is about the diabetes management, there was a great kind of personal story in there just dying to come out, where you could really almost pull on the heartstrings of the people you're pitching to make it really clear to them there's a big problem and you have a real personal interest in solving that. There are lots of different ways to achieve the sizzle, but ultimately it's expected that there's a little bit of sizzle, that we're going to be a little bit visionary, that we're going to talk about doing big things. But the stake matters too. And what I mean is that even if I am using words in my corporate pitch where I say things like next generation technology and fundamental improvement, and I and others can use even more lofty terms, of course. Revolutionizing is a favorite verb of our industry, for example. You're still going to be judged partly on whether that sort of opens the door. That maybe gets the attention of the people that you're pitching to, but ultimately you will be judged on delivery. And delivery just means, can you do what you say you're going to do? And that's not have you revolutionized medicine. That's have you defined a specific target and have you met it? So that could mean we aim to be in the clinic by 2024. Or we aim to have an IND filed by late 2022. Investors expect not to speak to you more than, they expect to speak to you many, many times. And I think that other than those who sort of chase momentum and milestones, what they look for in their repeated interactions with potential investment targets or actual investments is a consistency of message and a consistency of delivery. And on that point, I would simply say it's best to under-promise and over-deliver as much as you can. There's, despite the increasing precision in the scientific tools we have at our disposal, despite continued understanding and improvement of understanding of how best to develop drugs, for example, there's still, a lot of uncertainty, and mistakes can happen, and mechanisms of action don't play out. I think it's always just best to the extent possible, define for yourself what you think you can realistically achieve, and try to sell just a little bit less than that, so that when you deliver what you think you can achieve, you'll look like you have performed outsized, ahead of expectations. Does that make sense?
Yeah, thank you.
Yes, question.
Yeah, can you hear me okay?
So far.
All right, great. You mentioned the large pharma partnership with Vanda, and then obviously now there are partnerships with Genocea with like Oncovir and Novavax. Can you talk a little bit about your perspective on pursuing partnerships versus making the decision to pursue a venture individually? And then maybe like the challenges and also the benefits that come with partnerships.
Yeah, two good questions. And but they're not unrelated to each other. I think David said something earlier about sort of having a realistic assessment of what tools can do and kind of defining mechanisms of disease and finding targets. And I think a similar realism or humility is always going to be important in trying to advance your company. And I think the value of partnerships generally is that, and ultimately the best reason to do partnerships is because you believe that your partner can do things that either you can't do, or you don't want to do, or they can do much better, which could mean faster, cheaper, more capital efficient, we can do lots of things, than you. So I think that's the underlying thinking we have when we contemplate potential partnerships. I'll also say that we're not the only ones who think this way, and everybody I should say nearly everybody in the ecosystem, whether it's larger companies or smaller, are constantly assessing for themselves whether, for example, advancing a program or launching a program outside of the US is best something that they are in the best position to succeed or not. And there's usually some combination of a cold hard calculus that says we're going to be able to deploy capital better or less better than these guys, and so we should do deals. Plus, look, there's always going to be some degree of sort of the softer stuff as well that are ultimately just as important, ego and things like that. And so, that gets, I think, to your second question of how do you think through the pros and cons? The numbers, the analysis, the strategic rationale, and the financial modeling underpinning it matter. Certainly small companies, I think there's a little bit of a conventional wisdom that the more control smaller companies have, the better positioned they are to deliver long-term value for their investors. I think that analysis may bear out if you look at a portfolio of companies over time, but I think it dismisses the element of risk. I think that when we build these models that say, well, gee, if we can get, making this up, but if we can get somebody to give us $50 million up front and 15% royalty on sales, then it, puts us in a different funding position, a different competitive position, it brands us differently. Those are all good things. But if it doesn't, those numbers only make sense if you have a really good sense of your clinical, regulatory, execution risk. So I think when we think about partnerships generally, we're always thinking about the strategic landscape and such factors. We're thinking about the financials, but I think we also need to be thinking about sort of the end goal of our companies. And in the case of Genocea, we want to see cancer immunotherapies, sort of with the Genocea name, solving some of the toughest cancers in some patients who are really running out of options. That's our North Star, but that doesn't mean that we have to do it all by ourselves. We have to, absolutely value the things that we think we can do well in terms of working rapidly, adjusting, we hope, nimbly, being really laser focused on these patients and their needs, perhaps better than a larger company. But look, if another company can bring to bear the right combination of, in this case, cancer domain expertise and commitment to see what we think is important, so being really thoughtful about pricing and the commercialization model to guarantee as best we can that everybody who needs this therapy, assuming it's approved, of course, can get it. And of course the financial considerations as well. So I think that all of these sort of hard calculations as well as kind of broader strategic considerations are things we have to keep in mind as we think about partnerships. Have I answered your question?
Terrific question. We're a little short on time, unfortunately. One question I wanted to pose to you, Chip.
Yeah.
Statistics, epidemiology, advanced chemistry, specific lab techniques. What language or skill is toughest to find right now in your industry?
Toughest to fund?
To find.
Find. I would say statistics. I think that, we're not looking for statisticians specifically, but what I would say is that so much of what we can or should do and the way we're able to be convincing about it is based on designing good clinical trials and understanding how much certainty we can take from clinical trial results, depending on their design and size.
Boy, am I glad that was your answer. I've been focusing on that. Chip, I would gladly spend the entire rest of the day discussing this with you. And sure, 3/4 of the class would probably stay here too. Unfortunately, I gotta let them get to the next class. I cannot thank you enough for spending this time with us. It's been fabulous. Thank you so much. Thank you again. Hey guys, if you have, please sign out if you can. I'll get back to you during the weekend, next assignments. If you came to class unprepared to pitch today, please come prepared next week, because the likelihood is we will give you the opportunity to rectify that. And I'll be hanging around here in the classroom for the next hour or so. So, good class, thank you.
Such a good spirit. He answers everything so
Yeah, you can tell.
exactly. That's why he's not.
I can see the recording.